Hello, everyone. Thank you very much for coming. It's a really big pleasure to be able to talk to you today. And as it has already been said, I would love to introduce the first challenge of this year's edition CAMDA session, and also a brand new challenge which is happening this year for the first time, which is the gut microbiome based health index. So I'm sure that you've all heard about the microbiome. You probably listened to some other microbiome-related talks, but I would like to introduce the concept once again to just make sure that we are all standing on the same page. So what is a microbiome? A microbiome is, actually, let me just change this to a laser. So a microbiome, we'll be focusing specifically on the gut microbiome. It is a community of microorganisms, such as bacteria, archaea, viruses. They interact with one another and they interact with their host. There are a number of levels from which we can study the microbiome. So we've got the 16S RNA sequencing, and we also have the metagenomics. Those two methods allow us to identify the taxonomy, so search for the actual bugs that are present within in the samples. The 16S at the top is the first type of sequencing that was used. It is relatively cheap. It is based on the idea of a hypervariable region that is present in all bacteria. And it allows us to identify which genera of bacteria are present within our stool samples. The problem with it is that I already said it's only down to the genus level, whereas we know that the concept of species and strains is really important. And that's why we have the metagenomics, which looks at the complete DNAs of the bacteria. It sometimes even allows us to go down to the strain level if we have sufficient sequencing depth. And there is also another strong advantage to using it because it allows us to look at the functional profiles, which is not possible with the 16S-based sequencing. What this means is that we can look at the functions that the microbes have, the metabolites that they produce, the way they work within the community. And then we have three more levels, which is the metatranscriptomics, a study of the gene expression. This method allows us to look at the specific active processes that are happening within the system, because we know that the system will be contaminated with stuff that is full of dead bugs, for example, or contamination from the host. And then we have metaproteomics and metabolomics. Those are studies of the proteins and metabolites that the system possesses. So again, they are a way to identify those active processes and provide the link between the DNA-based analysis and the actual validation of this interaction with the host. So why would we want to evaluate our health from stool? You might have heard of the gut-brain axis. It is a phrase that tells us about the strong interaction that is happening between our brains and the guts. And it is a validation of how much our gut influences the rest of our bodies, our thinking, our behavior, the way we think. So it is a chicken and egg question whether the microbiome is changed in the disease or whether it is the disease that is causing an alteration in the microbiome. But we know that the microbiome does indeed change in disease, and hence we can use this information to track our health over time or to compare it between different individuals. Microbiome-based methods are a very good alternative to other tools, such as blood collections or biopsies, because they can be collected regularly, non-invasively, and they're really affordable. Who knows, maybe one day, hopefully, we will be able to collect stool and analyze it directly in our homes. So this is where the challenge comes in. As a part of the challenge, we provided 613 samples, metagenomic samples that were from the Human Microbiome Project 2 and 2 American Gut Project. So the samples included healthy people and people with inflammatory bowel disease, or IBD in short. We provided taxonomic and functional profiles. Those were already pre-calculated by us with the biobakery tools, Human and Metaflan. And we also pre-calculated health scores produced by different already existing health indices. So we used taxonomy-based Shannon entropy, the gut microbiome health index, so the GMHI, and its successor HiPCA, which is almost like the GMHI, but it produces some additional transformations to the data. So now it's your turn to develop your own index. We are really looking forward to hearing about your solution to this problem. And what I'm going to talk to you about in a second is why we think that we should move away from taxonomy-based indices, start moving towards functions. And we are also going to present you our approach so that you can beat it, maybe. Okay, so what are the best health indices to date? There are two classes. The first class of indices is purely based on alpha and beta diversity. So the idea behind those indices is that if you have a rich microbiome, if you have many different species, you tend to be healthier. And the second class is based on taxonomy. So those indices look at the ratios of the so-called beneficial to pathogenic bacteria, and depending on what ratio you end up with, you're healthier or less healthy. And this is exactly what the GMHI, so the Gut Microbiome Health Index, does. This index is one of the most comprehensive indices to date, and it was published in Nature Communications a few years ago. Actually, it's also included as a QIIME2 plugin. It was compiled within our institute, and it's freely available if you would ever like to use it. Okay, so why do we need a new index? Let's have a look at the recently redefined microbiome definition. So when we look at the definition, we can see that the microbiome consists of the microbiota, so all of the bacteria, viruses, fungi, etc., etc. But actually, the majority of the definition is the so-called theater of activity. So it is all the functions that the species have, all the metabolites that they produce, the specific environmental niche that they're present in, but also their interactions with each other and with the host. So in order to capture what is happening within the microbiome, we can't just look at this small part. It's the way the current indices do it, but we need to consider the whole microbiome definition. And here I'm going to show you a direct proof why using species or taxonomy-based indices is not a good solution. So the distributions you can see at the top is the distributions of species and functions in the healthy samples from the Human Microbiome Project 2. When we look at the distribution of species in the healthy population, we can see that the majority of species are found in just a small subset of individuals. So if you're trying to find species that would be universally beneficial and present in all of us, you just can't do it because we do not share the same species. But if you look at the functions, of course, there are many functions that are present in just a small subset of us, but the majority of functions will be highly prevalent. And therefore, why not base an index on those functions in order to develop a better index? As I mentioned, this was just the Human Microbiome Project 2, but the question is, do we see it in other healthy cohorts? And the answer to this question is yes. When we add new healthy individuals from the American Gut Project, we can see exactly the same trend. So there's a significant enrichment of functions which are present in over 80% of individuals. We therefore call them the core functions, and we based our analysis on this concept. Okay, so this is just a small look at what we actually did when we tried exploring what a healthy microbiome definition is. So here on the left, we can see, again, the Human Microbiome Project 2 and 2 American Gut Project cohorts. So this includes people who are healthy and who have IBD, so Crohn's disease, ulcerative colitis, and actually the American Gut Project 2 also had obese individuals. So the x-axis represents Shannon entropy based on functions, and then the y-axis is based on species. When we look at the distribution of those samples from the Human Microbiome Project, we could see that any classifier we built would be separating those samples quite well. So in this case, this data set is relatively simple to classify. But when we look at the American Gut Project cohorts, we can see that this trend is not obvious. It is very hard, if not impossible, to classify those samples. if we wanted to put one specific thresholds for all of those cohorts, it would be impossible, because we can see that, for example, we have a set of samples here in this range, whereas we do not see any samples at this position from the other American gut cohort. So we need a smarter way to classify our samples. So we use the concept of our co-functions. Actually, here we use the LEFC, which is a pathway enrichment approach. And we tried searching for functions that would be enriched in health or in disease. When we compared how those enriched functions were overlapping between different projects, we identified 100 functions that were associated with health, and then almost 200 functions that were associated solely with disease. Okay, so we had those functions, and we wondered what type of functions there were. So we looked at the content of course functions within those enriched functions. Actually, we didn't get any interesting results for the American Gut Project 1. We only had 10 enriched samples, and they hardly passed significant thresholds. So we focused mainly on those two projects. And we can see that over 90% of samples which were enriched in health were actually indeed our core functions. So we can see that those functions are highly prevalent in healthy samples, and they are enriched. And so we continued our analysis, and we ended up with a set of features that describe the healthy microbiome. We defined them. We actually ended up with 5 or 6 functions or characteristics. We trained a random forest predictor, and then we produce a health score, which is between the values of 0 and 1, where 1 indicates absolute health and 0 indicates disease. So in other words, we aimed to construct an index which would classify healthy from IBD individuals better than the already existing taxonomy-based indices. And those are our results. So you can see here at the top left, Shannon entropy, which is a measure of diversity. Then we have the GMHI and the high PCA, so the two taxonomy-based indices which look at the ratios of beneficial to harmful bacteria. And then we have our index. So again, we have 3 cohorts, the Human Microbiome Project 2 and 2 different AGP cohorts. And the green bars represent healthy individuals, and the other colors represent the dysbiotic individuals with IBD or obese individuals. So we can see that when we look at the Human Microbiome Project 2, our index is producing the greatest separation between the healthy and the diseased individuals. GMHI is doing a similar job, but actually Shannon entropy and high PCA are struggling a bit. When we look at the other cohort, again, high PCA and Shannon entropy are not able to distinguish between those individuals. And GMHI and our index are comparing, have a similar performance. And finally, the last cohort, when we look at the obese individuals, so those shown in brown, actually Shannon entropy, GMHI claim that obese individuals are healthier than people who are healthy, and our index is the only index to produce a drop. So we managed to show that our index is better at distinguishing between healthy and IBD individuals, but we wondered whether it was applicable to other diseases too. So we tested it, and the trick here was that all of the other methods had previously saw those data sets. So they were familiar with those diseases, and our index had never seen them, didn't know they existed. So we tested for diseases, colon cancer, acute diarrhea, ACVD, and the metabolic syndrome. And our index distinguished between healthy and colon cancer and acute diarrhea and healthy people quite well. Actually, we got the best separation here in the colon cancer cohorts in comparison to the GMHI and high PCA. ACVD turned out to be a bit more difficult to classify, though we had a good trend. And the metabolic syndrome was the most interesting, because when we look at the results, actually, GMHI and high PCA claim that people who are healthy are more diseased than people who actually had the metabolic syndrome. We are not producing a strong separation, however, it is at least of the expected direction. Okay, so this is where we end up with our test set that we provided as a part of the challenge. We provided 35 matched patient samples. They consisted of the healthy hospital staff and our Polish COVID-19 healthy cohort, COVID-19 disease cohort. Each of the pairs was sampled at 2 time points. The first one was at the day of admission when a person got admitted to the hospital with COVID. And then the second one was the last sample from the ward stay. When it comes to the hospital staff, it was just sampled at 2 random time points. So the question to the competitors was, can you identify the healthy samples? And then, can you say anything interesting about the COVID-19 cohort? So we said it deliberately because we put a trick in the data. So actually, the COVID-19 patients were divided into groups. This is something that the competitors didn't know about. The first group of individuals were people who recovered, so we would expect to see an increase of health at their second time point. And then the second group of patients were people who got much worse, and they were ultimately transferred to the ICU. So I'm really looking forward to seeing if anyone managed to guess this. So I'm now going to show you the predictions of our index on this COVID cohort. Actually, here we compared shallow and deep sequencing. Those were the same samples. And what we can see here is, again, green bars represent healthy people. And then let's focus on the brown bars. So the light brown color represents people with COVID who admitted to the hospital. And then the dark brown people who ended up in the ICU. So naturally, if someone goes to the ICU, it means that their health deteriorated, so we should see a drop in the index. And this is not what we observed with the GMHI and the high PCA, because they clearly show that the people recovered, which is not the case. And our index was the only one that produced this drop, so I was able to identify this longitudinal decrease in health. So we were curious whether those results were translatable to other to the deep sequencing. And they partially were. So the GMHI actually failed completely. It was not able to classify between healthy or disease, deeply shallowed. High PCA, again, claimed a completely inverse trend, claiming that healthy people were more diseased than COVID people. And our index was the only one that produced a separation and nicely showed that COVID people were more diseased than healthy people. So one of the last slides I'd like to show you is robustness to depth once again. This is another reason why using taxonomy-based indices is not a good idea. So let's have a look here. What we did here is we verified the data. So we tried simulating our deeply sequenced samples to different sequencing depth, so between 2 and 20 million, in order to see how the values of the indices change as we change sequencing depth. There were in total 50 GMHI species that were associated with health or disease, but when we actually look at our data set, we can see that across all sequencing depths, we only identified one GMHI species that was identified with health, and only up to 4 species identified with disease. So once again, the original paper claimed that there were 50 of them. So we couldn't see them in our data, therefore this index was not translatable. When we look at the index values for the GMHI and our index, I marked here with the orange line threshold for health. So when we look at the GMHI, actually it claims that people are healthy only at the level of 2 million sequences. But as we go higher with the number of reads, we can see that it's actually starting to predict disease. So depending on the sequencing depth, it will not be stable with its predictions. Whereas our index, actually the threshold for health is here. Regardless of the depth, we are still predicting health. And the last slide, it's comparison, gathered comparison of different indices to different diseases. So what we did here is we ranked the diseases according to their, the indices according to their importance. So this is our index in the end, and then all of the other indices. The higher the value, the more important an index. So for example, in this case, our index was the most important. and the classification of HMP2. And the IPCA was the worst. In the cases where it's written rejected, it basically means that the index values were not informative and the index was not working correctly. So our index was the best because it achieved the lowest mean of the ranks. So overall, it obtained the best classification on all diseases. It also got 2 rejects, whereas there were some other indices that got rejects, and it also won the most times. So we are happy about this. So yeah, we developed an index purely basing on the features describing a healthy microbiome. We targeted healthy versus IBD individuals, and we didn't include any other diseases in our work. However, it turned out that our index was nicely translatable to other diseases too. The work is still in progress. We would like to make it more individualized, focus more on the longitudinal data, because we believe that tracking your health over time and searching for any dysbiosis is more informative than by looking at cross-sectional data. And the big question is, can you beat us? Also, one technical note I would like to put here is that we have not provided the correct answers or labels for the COVID dataset, but we will be providing it soon via email in order for you to compare your results properly when you're writing up the results and submitting your papers. And I would like to thank everyone involved in the project, Dr. Paweł Łabaj, our whole lab, and of course all the collaborators. And I am now happy to take questions. Thank you.
No, it's okay, I think now. So please come here. So just 2 comments. As Kinga has said, the COVID data set has been just published. So the labels are now revealed and people can work further on this and to prepare the full papers after the CAMDA. And also the index by the work by Kinga is available as a preprint. So if you want to get either just look into the CAMDA Twitter, it should be somewhere there, the link, or just approach us and we are happy to share. And now, yes, please, the questions.
Oh, well, shoot, I might have answered my question, but let me ask again. So you were saying with regards to the test set, is that now unblinded? Is that publicly available?
Yes. So it will be now publicly available.
Okay. Well, shoot. Is there interest in having a blinded data set to continue this challenge? I think that could be a very useful resource to the community.
We're working on the iteration of the challenge for the next year, and then.
Okay, that's very good to know. I'll keep an eye out. Thanks.
Yep.
So, if anyone has questions. I have a gazillion questions, but I'm only going to ask one for the sake of time. There's bound to be batch effects that are specific between groups. So how can you know for sure that what you're picking up is specific to disease and not specific to the type of data you're dealing with?
Yeah, of course, there are a lot of batch effects. There are a few levels from which I was trying to answer this or cope with this problem. First of all, I tried making the features describing the microbiome that would be going into the random forest as universal as possible. So for example, I was not using absolute counts or anything like this. I was trying to look at the ratios, which would be universal. And also, so the stage at which this current index currently exists is that it is cohort specific. So we are only able to distinguish between samples at the level of a particular cohort. Also, this is something that it's also as a result of the batch effects and cohort specific characteristics. But what we are working on is trying to understand what a healthy microbiome is and how those cohorts differ from one another so that we can rescale the index ultimately and remove those batch effects.
Sorry, yes, of course, you want to focus on the cohort to get the results that you're getting, but in the end, the idea is to predict for novel data.
Yes. Yes.
So,
Therefore, we need to learn how to rescale the index accordingly, depending on the characteristics of the cohort. For example, based on sequencing depth.
you think you need to rescale the index and not the data?
It depends. It depends. Because there are
Okay.
different approaches, right, to doing this. You can either, so it's all about data, for example, pre-processing, like you're saying, in order to correct for the batch correction, but also it's at the level of the index, depending on the features that you're describing the data set with, you might need to scale them accordingly.
Okay. Thanks.
Okay, so one more question, then we'll run for the next.
Hey, yeah, this was really interesting. There's some, meta-analysis results that suggest that maybe like IBD is like a disease that's characterized by like loss in the microbiome, whereas others might be characterized more by gain, right? Like, I'm trying to think of an example, but I think colorectal cancer, like, you tend to have like a gain in non-health associated species, right? And since you were focusing on kind of like, the health associated species, I wonder if it would generalize equally well to, whether COVID was more like IBD or more like colorectal cancer, and then like whether it would generalize to a case like that.
Yeah, so this is exactly a question that we are trying to answer now.
Yeah.
As I mentioned in the beginning, we were focusing specifically on HERV versus IBD individuals, and it just turned out to work on other diseases. But what we are working on now is searching for those differences in specific diseases so that we can then make it more applicable to a wider range of problems.
Makes sense. Thanks.
Thank you.
Okay.